Welcome and thank you for tuning in to Queensland Brain Institute's MND Breakfast at Home Edition, a QBI update delivered a little bit differently. My name is Rebecca Sandgill and I am the current MND Early Career Research Fellow and a member of Dr. Adam Walker's lab at QBI. The COVID-19 situation has prevented us from gathering together and holding our annual MND breakfast, but we didn't want to miss the opportunity to keep you informed of the progress being made in the fight against MND during MND Awareness Week. Today, I'm joined by Dr. Adam Walker, Dr. Shuan No, Professor Trent Woodruff, Professor Naomi Ray, and Professor Peter Nesta, all from different areas of UQ, and they will share their work in MND research and also what they hope to ultimately achieve through their research. I'd like to tell you a little bit more about motor neuron disease, also known as MND. As many of you will know, MND is a rapidly progressing neurological disorder that attacks motor neurons in the brain and spinal cord, controlling the way we talk, eat, speak, and breathe, and walk. The loss of these motor neurons leads to progressive muscle weakness and wasting. And currently, over 2,000 people are living with MND in Australia. This destructive neurological disorder currently has no cure and no effective treatments. UQ and the Queensland Brain Institute are using a multidisciplinary approach to improve the understanding and treatment of motor neuron disease. So today I will begin the update by discussing my research. I've recently commenced a MND Early Career Research Fellowship, and as I mentioned, I work in Dr. Adam Walker's lab. The focus of this fellowship is to screen human genes to find the molecular on switch for motor neuron disease. So if we took a magnifying glass and if we look in the brains of people living with MND, what we would find are these proteins forming really large clumps inside of the motor neurons. We believe that these clumps may stop neurons from performing their normal functions in the brain and spinal cord, that is, innervating muscles and allowing us to move our arms and legs. And when these neurons are stopped from performing their normal functions, this could lead to their death in a process called neurodegeneration. Therefore, we are searching for new ways to stop protein clumping as a therapeutic strategy to treat motor neuron disease. We are using the revolutionary gene editing technology called CRISPR, you might have heard it in the news, to scan every single human gene. That's around 20,000 genes. It's a lot of genes that we can investigate all at the same time. And in this way, we aim to identify the genes that both trigger and protect against this abnormal protein clumping. This technology will enable us to fast-track research and also to let us focus on a small number of genes. And our ideal gene would encode a protein that can stop the formation of these clumps, but also enhance and extend the quality of life of a neuron in the brain. What's really unique about our lab is that we have the expertise to both study what's happening on the molecular scale, but also then translate those findings into preclinical testing in an animal model of motor neuron disease. And using this strategy to find viable therapies for people living with MND is the absolute key objective of our research. So I'm just going to answer a question that was sent to us. The question we received is, how long do you believe it will be until we have a cure for motor neuron disease? This is a really curly question. It's very difficult to estimate the actual time that it will take to find a cure for motor neuron disease. But I do want to say that there are thousands of researchers across the globe working together and working towards a cure for motor neuron disease. The motor neuron disease research community is extremely well connected, and as a consequence, we now know many of the events that occur at the start of disease at the cell and molecular level. So now it's a matter of identifying from all of that research, what are the most promising cellular pathways or combination of pathways that we should target with drugs or with gene therapies or with gene editing therapies like CRISPR. So I'll leave it there. And as I mentioned, I work in Dr. Adam's lab. So I think it's a good chance now to hand over to Adam so that he can discuss his current research. Thank you.
Great. Thanks very much, Beck. And that was great to hear about what you're working on. So I'm the Ross McLean Senior Research Fellow and the head of the Neurodegeneration Pathobiology Lab at QBI. I've been at QBI for about 2 years now, and I lead a group of researchers that are all dedicated to understanding the causes of MND. I wanted to start by talking about my position before I start talking about my research. So my position at QBI is supported by lots of generous donors, and it's named in honour of the late Ross McLean, who sadly passed away from MND in 2005. Even while Ross was battling MND, he and his family and friends decided to try to develop a way to help research into the disease so that one day we'll have a cure for MND. This effort's been led by Ross's son, Jeff McLean now, and there's been numerous fundraising events in the community that have been in support of the Ross McLean Fellowship. So I'm really honoured to have that title now in my research. So the research in my lab is really aimed at trying to understand the very early pathological changes that occur in motor neuron disease. So we do a lot of studies in mice that develop MND. And from the mice, we can look very early on at the biochemical changes that are occurring in the brain and the spinal cord, even before they develop symptoms. And that's really something that's really hard to do in people. So if we can understand exactly why specific types of nerves are degenerating in motor neuron disease, then our theory is that we should be able to devise really targeted strategies to develop specific drugs or gene therapies for people with MND. So that's the goal of what we're working on, is trying to find ways to stop the disease for people living with MND. Beck, do you have a question from the public?
I do, yes. How do you give a mouse motor neuron disease, and is a mouse with MND similar to a human with MND?
Okay, yep, that's a really good question. So the goal of using mice in our studies is really to recapitulate the pathology that we see in the nerves of people who have motor neuron disease that we can only see after they've passed away and we've been able to do an autopsy on their brain and spinal cord. So the way that we do that in the mice is to use genetic engineering. So that means that we specifically alter the DNA in the mice so that the nerves accumulate the same pathology that we see in people with motor neuron disease. The benefit of using a mouse is that we can make specific changes to their DNA and then study how that affects them over time. And what we do see in mice when we do this is that they do develop a progressive disease, which is similar to motor neuron disease. So they have a gradual decline in the ability to move, and they show a decline in their muscle function over time. So when we look also in their brains and their spinal cords, we can see that they do have this pathology that we see in people with MND. Obviously, studying another animal that isn't a person is not perfect, but I think that we're at the point now where we have quite good mouse models that allow us to study the disease. And as I said, it's really helpful to use mice because we can look at their brains and spinal cords early on in the disease process.
Thanks, Anna. That was really interesting how you're working with the mice to understand the changes throughout the disease. Next, we'll pass over to Shu. She's just commenced a new fellowship. So Shu, explain a little bit about the fellowship and your current research. Thanks.
I'm currently the FIGHT MND Mid-Career Research Fellow. I transitioned into this fellowship early this month. Prior to this, I was the Scott Sullivan MND Research Fellow. And what's really special to me is that both of these fellowships really are, I guess, in memory of two fantastic people who lived with MND and who have since unfortunately passed away, but who really wanted to leave a legacy behind in continuing the fight against MND and looking for this cure for this disease. So I am, as I said, the FIGHT MND Mid-Career Research Fellow, and I run a laboratory that is based at the Australian Institute for Bioengineering and Nanotechnology, but I'm also an affiliate with the Queensland Brain Institute. The research that our lab does also crosses over to the Royal Brisbane and Wesley hospitals. Our research is really focused on understanding how changes in the way the body or cells use energy might impact how quickly MND progresses from one person to the next. We've been doing this research for the last 5 years. And most recently, we've identified a few key changes in muscle and neurons that we think could be targeted as a potential treatment to help slow MND or to eventually stop MND in its tracks. And because of these discoveries in the lab, we have more recently been testing a few compounds that we think might target these processes. And we've recently published a manuscript using mouse models of MND where we've shown that one of these compounds is actually very effective in slowing the disease, albeit very transiently. So we've learnt a bit more from this recent paper, and we're hoping to take this into the next trials that we are doing in the lab. So eventually, with all this drug testing that we're doing in the lab, we hope to transition these compounds into trials for people living with MND. And fortunately enough, we have received some funding from Fight MND to potentially start a trial of one of these compounds later this year.
Thanks so much, Shu. That's really exciting work, particularly moving into human clinical trials. I was just wondering, what does a typical day in the MND clinic look like for you? Because Adam and I are very lab-based. We work with cells and we work with the animal models of MND. So what does a day in the MND clinic look like for Shu?
I think that's a tough question, actually. A day in the MND clinic is never the same. And I think that's very much the same in the lab, right? No day is the same. And that's why we do research. We're very curious and we're at the forefront of discovery. One thing that's, I think, really great about working in the clinic, and I think being in touch with people who are living with this disease every day, is that you really begin to appreciate how different MND really is between people. People always say that no one person is the same, and you don't really see how different people are until you meet many, many people who are living with this disease. And it's through meeting all these people in Queensland and even interstate who are living with disease that we can learn more about MND. And it's learning more about MND and the person, which is, I feel, what's going to help us find a treatment, because we can't, I believe, target just one specific thing in the disease. We're going to have very personalized approaches. And what's also really important about working with people in MND is that they don't just contribute to my research by, coming every 3 months to see us and donating tissue samples and blood samples for research. The time that MND patients give to us and the samples that they give to us are actually used by a number of researchers in Australia and in Brisbane and also across the world. And one of the people who do use some of these samples is Professor Trent Woodruff at the School of Biomedical Sciences. And he's doing some really exciting work into the immune system. And I'll pass on to him so that he can discuss a bit about what he's doing.
Thanks, Shu. So hi everyone. I'm a pharmacologist by training, and I have a background in drug development. So we try to develop drugs to treat diseases. So our research group at the moment is really major focus of our group is to develop new drugs to treat MND. And as Shu mentioned, our research focuses on the immune system. In fact, a specific component of the immune system called the innate immune system. And there's actually a lot of evidence emerging that our immune system and this innate immune system that are normally active to protect us from infection are actually switched to hyperdrive, so to speak, in patients with MND. And a major effect of this increased immune activation is inducing a process called inflammation. So we're all familiar with inflammation and fighting infections, but our research has shown that there's increased inflammation in MND brains and spinal cords in mice. So as Adam pointed out earlier, we can study the brains and spinal cords of mouse models of MND, but this is just a mouse system, and it's kind of difficult to study this process in the brains of living subjects with MND. So to support some of the mouse work that we've been doing, as Shu mentioned, we've also now been very active in collaborating with lots of people at the Royal Brisbane Women's Hospital. And this has enabled us to collect some blood samples from patients with MND and also healthy volunteers. And we're examining the immune cells and the inflammation biomarkers in these patients to help us determine if what we're studying in the mouse is true in humans, but also to correlate some of these inflammation markers with the different presentations of the disease in humans. So as I mentioned before, we're a drug development lab. We've been developing a number of compounds which we think can block many of these immune proteins and hopefully halt that inflammation process. So we collaborate quite closely with a lot of medicinal chemists. It's important that the drugs we discover are able to be administered and enter the brain and don't have any toxicity. And this really helps us, these collaborations with these chemists help us to identify the best compounds for this purpose. So I guess most importantly, we've now shown some very encouraging results with some of these drugs. In the mouse models of MND. So we've tested the drugs in a couple of different models that we have at the University of Queensland with help from Adam, and we're showing some very encouraging results. And this has enabled us to now progress some of these drugs towards the human clinical studies. So as I said, the next work is now to take some of these drugs into humans. We've actually partnered with a few pharmaceutical companies to help with this. One of them is a Queensland-based company called Alcinex. And with support from Fight MND, we've now completed all the necessary preclinical and toxicology studies that you need to pass the regulatory standards that the Drugs Administration, the TGA in Australia and the FDA in the US, that what they require to enable human clinical studies. So we've now recruited some healthy volunteers for our early phase trial. It's called a phase 1A trial, which is a safety trial. So these healthy volunteers will be receiving drug in the next month to determine drug concentrations in patients who administered the drug and also to ensure that there's no toxic effects. And following this, we'll then begin testing the drug in patients with MND. So ultimately, what we hope to achieve from our work is to really identify new therapeutics that can be used in patients with MND, hopefully to slow the progression or help with some of the symptoms of the disease, or perhaps even to halt the disease in its tracks.
Thanks so much, Trent. So obviously there is an immense amount of work that goes from a positive result in a mouse to developing and executing a human clinical trial. I'm wondering if you could let our audience know how many months or years that development takes.
Well, as you say, it is a long process, and I think it's underappreciated timeline when it takes from getting an initial discovery in a mouse model all the way through to human testing. So the time frame varies depending on, I guess, the drug. If it's a brand new drug, the time can take a bit longer because you need to validate that it's safe for use in humans. Sometimes you can circumvent this if you take a drug that's an existing drug. If you pass through those preclinical hurdles within a couple of years, then you can actually make that process, say, within 5 years from initial drug discovery to testing in human patients. And then it can take another up to 5 years to actually get all the way through to approval and use in humans. So it's up to a 10-year process. So I think part of what we do in our lab is really focused on these immune proteins. And one thing we're very interested in looking at further is some of the mechanisms that are driving this activation of this immune system. And to help us with that work, we've been collaborating with Professor Naomi Ray, who's been helping us identify some of the genes, some of the genetics that are behind some of these pathways. So I'll pass you over now to Naomi, who can hopefully help talk to you about some of these processes.
Yes, thanks, Trent. Yeah, so I'm Naomi Ray. I hold joint appointments at the Institute for Molecular Bioscience and the Queensland Brain Institute at UQ. The fundamental question that underlies my research is, why are there differences between people? For example, why are some people tall? Why are some people short? Why do some people get disease and some people don't? And within those affected by disease, why are the symptoms and progression so variable between people? These questions are complex, and the answers likely involve both genetic and non-genetic factors. I focus particularly on the genetic factors, as these give clues for other biological research. So in my research program, I actually study many diseases, and when I joined UQ, I was asked to also study motor neuron disease. So to start with, we know that for about 5% of people diagnosed with ALS, they come from families with many affected family members, which is a very strong clue for DNA involvement. For many of these families, the DNA cause has actually been found, usually a single DNA letter that is actually different in those affected family members compared to everyone else in the world. We've learned a lot about ALS by studying these families, providing the clues which have underpinned much of the research you've heard about so far. However, my research focuses on the 95% of people with ALS who don't come from such families. For these people, there's still likely to be an underlying genetic susceptibility to ALS, which then compounds with other risk factors and then which together lead in some unknown way to disease. The genetic contribution here is likely to be complex, involving many DNA variants. And for these people, we can't learn much about the genetic contribution by studying their family members, because we know that the family members hardly ever get affected. Instead, it's really the particular combination of DNA letters that affected people have. So to learn about the genetics of this common type of ALS, we need to actually contrast the DNA of those who do get ALS to study samples. So when I joined UQ, I realized that although some ALS research clinics in Australia were collecting DNA, a lot of people with ALS were not actually being given the opportunity to contribute to this sort of research. We received funding for 3 years, actually from the Ice Bucket Challenge, which was awarded through the Motor Neuron Disease Research Institute of Australia. So although my interest is what we can learn from the DNA, we need as much information about people to really disentangle all the contributing factors. Success of the SALSA project helped bring in actually additional funding from the National Health and Medical Research Council. In addition, FIGHT-MND liked what we'd done. FIGHT-MND recognised that the SALSA project was building up an important data resource for research, and that many people with ALS are very pleased to be able to contribute to research in this way. FIGHT-MND recognised that the clinical trials often have inclusion-exclusion criteria which are actually based on genetic information, and this information is needed prior to the start of the clinical trial. Also, it's important to make sure that people across, so with funding from FIGHT-MND and also working closely here in Queensland with MND and me, who in turn work closely with MND Queensland, we're trying to ensure that everyone in Australia with ALS has the and also, just to be clear, there are many people who have less common forms of MND, such as PLS, flail arm subtype, etc. Our biological sample collection includes all these. And last, to understand the genetic and non-genetic risk factors that are associated with the motor neuron diseases, we need to make contrasts with people not affected. So in June, we're actually planning to launch a community recruitment. The last 10 years has actually seen an incredible advances with what we can measure from a simple blood sample. And so this gift is really worth its weight in gold.
Thanks, Naomi. That's really interesting, and your research has been really important to bring together sporadic ALS research in Australia. So I was wondering, if you need such large sample sizes to draw robust conclusions from your work, how do you collaborate and who do you collaborate with around the world to share your ideas and research?
Oh yes, great question. Yes, absolutely. We work very closely with research teams around the world. For example, our first samples collected under the SALSA project are being analysed together with 20,000 samples from around the world, and when put together have identified ALS risk genes which seed into many other areas of research. In addition, we've made sure that our data collection measures align very closely with those collected in other countries, which really helps future-proof the data resource, ensuring that we can integrate things down the track. In fact, one of the measures that's perhaps the most complex to track are the cognitive changes that occur for some people during the progression of their disease journey. And this is where the research of my colleague, Professor Peter Nestor, is so important. So I'm going to pass over to him to let him tell us about that. Thank you.
Hello, my name is Peter Nestor. I'm a neurologist, and I actually come to motor neuron disease research through the back door in a way, because my area of expertise in neurology is actually what's called cognitive neurology, or dementias. A proportion of people who develop motor neuron disease will also develop a form of dementia known as frontotemporal dementia. And that's an area that I've worked a lot on over the years. And one of the areas that I focused on with that is that frontotemporal dementia is not one pathological process. There are completely different diseases that cause this same clinical problem. Some of them associated with what's called TDP pathology and others with tau pathology. And it's the TDP one which overlaps with motor neuron disease. And so in the course of my work, I do a lot of brain imaging work trying to find new ways of saying who precisely has which disease during life. And because of that development of new MRI, magnetic resonance imaging techniques to try and get at this, I started to focus more and more on motor neuron disease, on the motor disorder as well, not just on the dementia, through the developments we were doing of trying to develop better ways of saying what's going on in these diseases. So what you can see here in this slide is, up the top, is a pathological specimen of a spinal cord of someone who's died from motor neuron disease. And the spinal cord's of particular interest in motor neuron disease because it contains both the wires, if you like, coming down from the brain that are going to tell the motor system, the muscles, to move. And that's these areas on the sides here. You can see that in motor neuron disease, they've gone very pale, they've degenerated, as opposed to this area, which is the transmitting sensory information back to the brain, and that's preserved, and that's why it's nice and dark. And then the second thing that happens in motor neuron disease is that the cells themselves within the spinal cord that will then pass out and supply the muscles also degenerate. So that makes the spinal cord a very interesting area to be interested in motor neurone disease, because both what we call the upper motor neurone system, the messages from the brain, but also the lower motor neurons, the cells that are going to activate the muscles, are both in the same place. And understanding how these interact to cause the disease and what comes first and how it evolves would be terribly interesting. The other side effect of this, though, is that if we can do this in a very good way, we can also use it to make better diagnostic tests to say who has motor neurone disease at an earlier stage. So in an ordinary MRI scan, if one goes to a hospital and has a scan of their cord, one can see the cord very well, but there is no anatomical detail in it at all. It just looks like a big grey blob. And one of the techniques we've developed a whole array of them, we can now image the actual grey matter part of the cord, where those cells are that are going to pass out to the muscles, but also see the tracts. And what we've found with this is that we can show now very significant changes in both the pathways coming down from the brain in motor neurone disease and also in these anterior horn cells. And so this is still a work in progress, but given these early results, I think we can optimize this further, where we may even be able to see these changes in individual patients with the disease. And the big advantage of being able to do this, as opposed to pathology, is that we can do it longitudinally. So we can actually show how the disease evolves, which gives us something to test treatments against, gives us potentially a new diagnostic tool, but most importantly, it could give us insights into the sequence, the cascade of events that actually goes wrong that causes motor neurone disease, which could then be used to develop therapies in the future. So that's the kind of work we're doing, and at that point I'll hand back to our Master of Ceremonies.
It's really interesting, very beautiful MRIs there, so much detail. I was wondering, does your research in MND give you insights into how you can approach the imaging of other neurological diseases, or is it the other way around?
Well, it goes both ways, and that's precisely how this started. So, for instance, we talk about these different diseases. The major neurodegenerative diseases that affect humans are the ones that cause dementia, such as Alzheimer's disease, motor neurone disease, obviously, and Parkinson's disease. And then there's a whole lot of other ones that are a bit less common. And people don't walk into clinic and they're a clear-cut example of any particular disease. These are progressive conditions that come on slowly and can be hard to diagnose in the very early stages. So yes, a motivation of all of this work is to develop ways of being able to say much more accurately right from the start who's got what. So yes, this is relevant to all of the degenerative diseases, which is why I'm now taking this approach with my research of deliberately targeting everything all at once to be able to sort them out rather than focusing in on one particular disease at the expense of all the others.
Thank you very much, Peter. And thank you very much to all of our panel members for today, Adam, Shu, Trent, Naomi, and Peter. I think it's very clear from today's research update is that we're all working together, but we all do have very clear strengths in the research that we undertake. And together, we're going to forge forward to, realize a life without MND in the future. We hope that our audience has enjoyed hearing how MND researching is progressing at UQ and what we hope to accomplish through our collective efforts. And I would also like to take this opportunity to thank MND, who supports a number of our research projects, MND and me, and also all of our other wonderful donors who are the reason that we can continue our research, but also discover new ways to try and combat motor neuron disease. Please feel free to share this update with anyone who may be interested in learning more about motor neuron disease research. And if you would like to keep further informed about other scientific discoveries at UQ and QBI, please follow QBI on Facebook, YouTube, and Instagram, and we also have a monthly newsletter. So thank you again for joining us. I hope you have enjoyed our MND update.